Okay, I've got everybody from the waiting room joining, so I think you can get started whenever you're ready.
Fantastic. Thank you so much.
Mhm.
So hello, everyone, and welcome to the first session of the Association for Research in Otolaryngology seminar series on the topic of auditory nerve, brought to you by the ARO Education Committee. Today, we are delighted to welcome Professor Charles Lieberman from Eaton Peabody Laboratories at Mass Eye and Ear and the Department of Otolaryngology, Head and Neck Surgery at Harvard Medical School. I'm Dr. Kirupa Suthakar, Assistant Professor in the Department of Speech, Language and Hearing at the University of Texas at Dallas. I'll be your moderator today. If you have any questions during today's presentation, please use the chat function and direct them to me directly as the moderator to avoid interrupting Dr. Lieberman during his talk. Following the presentation, please feel free to raise your hand and I will call on you to unmute so that you can ask your question directly. Closed captioning is available and the session will be recorded for later viewing via the ARO YouTube page. So Dr. Lieberman is the Harold F. Schucknicht Professor of Otolaryngology, Head and Neck Surgery at the Harvard Medical School, and the former director of the Eaton Peabody Laboratories at the Mass Eye and Ear from 1997 to 2022. He received his BA in biology from Harvard College in 1972 and his PhD in physiology from Harvard Medical School in 1976. He worked with Nelson Kiang on the responses of single auditory nerve fibers in normal and noise-damaged ears. Following his thesis work, he did a postdoctoral stint with Sanford Pele in the anatomy department at Harvard, performing serial section ultrastructural analysis of the normal synaptic connections between auditory nerve terminals and inner hair cells. He's published over 215 papers on a variety of topics in auditory neuroscience and has received a number of awards, including the Award of Merit from ARO in 2009, the Baker Shea Silver Medal from the Acoustical Society of America in 2012, and the Grand Prix Scientifique from the Fondation pour l'Audition in 2022. Really sorry for butchering that. His research interests include 1, the coding of acoustic stimuli as neural responses in the auditory nerve, 2, efferent feedback control of the auditory periphery, 3, the mechanisms underlying noise-induced and age-related hearing loss, 4, the signaling pathways mediating nerve survival in the inner ear, and 5, the application of viral and drug-based therapies to repair the damaged inner ear. So on a personal note, I first had the pleasure of interacting with Charlie when we invited him to present at the Australasian Auditory Neuroscience Workshop back in 2014. Given my interest in auditory efferent neurons, I had read many of his early papers and was blown away by his clarity of thought and passion for the auditory system. Charlie agreed to take me on as a postdoc, and 2 years later I moved to Boston. I was really privileged to learn to record from single auditory nerve fibers in mice with noise-induced cochlear synaptopathy under his tutelage. Charlie is a rigorous scientist and phenomenal thinker. His observations and discoveries have revolutionized our field, yet he has remained humble and generous, taking the time to teach, mentor, and collaborate with many of us here today. So thank you for agreeing to present at this first session of the Auditory Nerve seminar series. Without further ado, please take it away, Dr. Lieberman.
Well, thank you very much, Kirupa, for that nice introduction. It's a pleasure to be here. As you can see, the title of my talk is Fundamentals of Normal Structure and Function in the Auditory Nerve. I'll be breaking it up into 4 chapters, as you can see here. And the first one, I'll talk about the basic wiring diagram, including the distinction between type 1 and type 2 spiral ganglion neurons. So here I show you a photomicrograph of a cross-section through the auditory nerve of a cat taken very near where it enters the brainstem and the cochlear nucleus. At this level of resolution, you can't see very much. You can see some white space, which are the blood vessels. But when I look at it, what I see is this general organization, which is reflecting the fact that the entire auditory nerve bundle made up of 40,000 fibers is tonotopically organized. as the nerve trunk exits from the apex toward the cochlear nucleus, the successive turns kind of wrap around the core as in this jelly roll, such that there's a tonotopic organization like this. At this low mag, you can't make out very much, so if we focus in on one of these fascicles, as they're called, you can see a fascicle here of auditory nerve fibers is made up of a couple of hundred elements like this. Each one of these little dark things is a myelinated nerve fiber. If we zoom in at even higher power, which requires going to the, that's moving ahead, which requires going to the electron microscopic level, we can see the myelinated type 1 fibers and an occasional interspersed very small fiber, which is unmyelinated, which is the central axon of a so-called type 2 auditory nerve fiber. This schematic here shows us a lot more detail about the distinction between type 1 and type 2 neurons, which were originally named and differentiated based on the shapes of their cell bodies shown here. As you can see here, as I've schematized here, and as I point out in these bullets, type 1 fibers contact inner hair cells and comprise 95% of the cochlear nerve, whereas the type 2 cells contact outer hair cells and comprise only 5% of the fibers in the auditory nerve. Importantly, as I alluded to just a moment ago, the axons of type 2 cells are unmyelinated, both the peripheral axons, as we call it here, and the central axons going to the cochlear nucleus, whereas the type 1 axons are myelinated. The type 2 peripheral processes go out to the outer hair cell region. They're highly branched, contacting many outer hair cells, whereas the type 1 are unbranched, for the most part, contacting a single inner hair cell. From these pictures in the center, you can see that the type 2 cell bodies are pseudomonopolar. They immunostain with peripherin, and back in the day, we differentiated them with silver stains because they also stain very darkly with silver stains. They both send central axons to the cochlear nucleus. We're not going to talk about the central projections today. And the other important factoid is that within the organ of Corti, all the neural processes of both type 1s and type 2s are unmyelinated. It turns out that the type 2 response, the type 2 cells have no response to sound, and the only way they've been recorded from successfully is to present electric shocks here in this study by Chris Brown a number of years ago, where he presented shocks to the cochlear nucleus and recorded from the cell bodies in the spiral ganglion. The axons are too thin, as you saw, to record from by micropipettes. So the only successful place to record from the type 2s is, at least in vivo, is in the ganglion cell region. As you can see here, when he recorded from type 1 neurons that respond to sound, they have very short latencies to shocks, whereas every once in a while he found a fiber that didn't respond to sound and had a shock latency of closer to 6 milliseconds. Much more recently, folks in the Glowatsky and Fuchs laboratories have recorded from the peripheral processes of type 2 neurons in vitro and have found the interesting results that they depolarize when the outer hair cells are damaged. And this is obviously strong evidence for an old idea that these unmyelinated type 2 neurons are actually nociceptors whose purpose is to tell the central nervous system when there's damage to the auditory periphery. So that's all I'm going to say about the type 2 neurons. The rest of this talk is going to be about type 1 neurons. So as is schematized here, with a couple of inner hair cells and a few unmyelinated peripheral terminals of type 1s, each type 1 fiber contacts a single inner hair cell by a single unmyelinated terminal. In this schematic here, we see that this unmyelinated terminal has a swelling at the end, and there's a region shown in light blue where it intimately contacts the inner hair cell. And there's a region in dark blue that we call the active zone, which is where presynaptically the voltage-gated calcium channels are present, and postsynaptically the glutamate receptors are present. And on the inner hair cell side, there's a structure called the presynaptic ribbon. Here I show some serial sections through one of these presynaptic ribbons. This is the inner hair cell side. This is the nerve side. You can see this prominent postsynaptic membrane thickening, a little less prominent presynaptic membrane thickening. From the serial section reconstruction, you can see that the synaptic ribbon is kind of like it's tubular, it's kind of like a hot dog, and it's surrounded by a halo of vesicles. This is the entire synapse at one auditory nerve fiber in a cat. And there's a surprisingly small number of synaptic vesicles tethered to this synaptic ribbon, on the order of just a couple of hundred. So seeing the synaptic vesicles on the inner hair cell side obviously is consistent with the idea that information moves from the inner hair cell side to the nerve fiber side when these synaptic vesicles fuse with the presynaptic membrane. A number of years ago, Joe Nadal showed that the synapses at the outer hair cells look interestingly different. There are many fewer synaptic vesicles around the synaptic ribbon, and there's often paired with them a structure which looks a lot like an efferent synapse in the sense that there are vesicles on the neuronal side and a postsynaptic cistern on the hair cell side. So this anatomical curiosity has suggested that there's bidirectional signaling at the type 2 synapse, which raises at least the possibility that in this highly branched type 2 peripheral process, there's actually local signaling going on where outer hair cells can talk to each other. But that at present is a highly speculative idea. Now, as most of you probably know, these synaptic vesicles are full of glutamate. There are voltage-gated calcium channels in the presynaptic membrane and AMPA-type glutamate receptors in the postsynaptic membrane at these type 1 synapses. A number of years ago, it became clear that you could beautifully immunostain these synapses using antibodies against a protein called CTBP2, which is a prominent protein in the presynaptic ribbon, and antibodies against glutamate receptors A2 and or A3. So in this confocal image here, you can see a row of inner hair cells with these opposed red and green puncta, each one of which is a synapse with an auditory nerve type 1 terminal. What's interesting in this picture is how different the appearance is in the outer hair cell region in the adult. These are from a mouse where there is no labeling with the glutamate receptor type 2 or 3. So another way in which the outer hair cell type 2 synapses are fundamentally different from those of the type 1 synapses with inner hair cells. So that's all I'm going to say about the basic outline of the wiring diagram of the type 1s and type 2s. So now we're going to move on to chapter 2, which talks about characteristic frequency, spontaneous rate, and the dynamic range of hearing. So this schematic reminds us that each auditory nerve fiber has a cell body, the spiral ganglion cell in the spiral ganglion, a myelinated peripheral axon, and a myelinated central axon that goes to the cochlear nucleus. When I started out in the field, I spent a lot of time putting micropipettes into the auditory nerve of anesthetized cats to record from the central axons of single auditory nerve fibers. And when one does that, one finds that each fiber is very sharply tuned. So these are classic tuning curves where we plot the threshold for response is a function of frequency. Here are two tuning curves from one auditory nerve fiber and another couple tuning curves from another auditory nerve fiber. At low sound pressure levels, they're very, very sharply tuned. And these tuning curves define characteristic frequency, or CF, which tells us where along the cochlear spiral this fiber is innervating inner hair cells. This light blue one, obviously more apically located than this darker blue one, more basally located, given that we all know that the base of the cochlea is tuned to high frequencies and the apex of the cochlea is tuned to lower frequencies. So in the late 70s and early 80s, we were able to apply techniques that people had developed in other parts of the central and peripheral nervous system to iontophoresis an enzyme called horseradish peroxidase into these central axons so that we could record from single fibers of the auditory nerve, pump them full of horseradish peroxidase, wait 24 hours for that protein to percolate through the central axon to the cell body and to the peripheral axon out to the hair cell, and through the central axon also to the cochlear nucleus. In the ultimate material, we could remove the organ of Corti as a microdissected whole mount, do the proper histochemical reactions, and we could find the very fiber that we recorded from. Here's a row of inner hair cells. You can faintly see their nuclei. The outer hair cells are out here. And this is the unmyelinated peripheral terminal of a fiber that we had recorded from and measured its physiological characteristics. So being able to do that allowed us to create a highly accurate cochlear frequency map. So these are the results of these single fiber labeling experiments in, I think, 15 or 20 different animals. And you can see that there's an extremely regular relation between log characteristic frequency and percent distance from the base. It's not quite a straight line. It kind of bends over near the top, and it looks a lot like what people call Greenwood functions, named after the guy who guessed at what this cochlear frequency map was based on psychophysical data. So although each inner hair cell, each type 1 fiber, I should say, is unbranched and contacts a single inner hair cell, each inner hair cell is contacted by numerous auditory nerve fibers, type 1 auditory nerve fibers, as schematized here. Here I summarize data from a lot of different studies, some of ours and lots from other people, where as a function of distance from the cochlear base, we plot how many different fibers synapse with each inner hair cell. You can see in general, they're sort of inverted U-shaped functions where somewhere in the middle of the cochlea, there's the highest innervation density with lower innervation densities toward the apex and toward the base. Interestingly, human here shown in the black and dark gray for adults and juveniles have one of the lowest innervation densities of all the mammals that have been studied, obviously lower than the record holder here, which is the cat, which can have up to 27 different auditory nerve fibers contacting a single inner hair cell. So what's the significance of all these different fibers innervating a single inner hair cell? Well, there are a lot of reasons why that might be important, but one is that it adds dynamic range. So in this plot here, each of these gray points is the tip of a tuning curve. That is, it's the threshold at the characteristic frequency of a different auditory nerve fiber. In this particular plot here, these are all fibers that were recorded from a single animal. One of the reasons why cat was such a popular experimental model for many, many years is that it was could be maintained very stably under deep anesthesia for actually days. And that allowed one to record from many fibers in a single animal. So in this particular case, I think there are about 350 single fibers recorded from one animal. So you can see that the most sensitive fibers at different regions of characteristic frequency have thresholds near 0 dB SPL, which of course is defined as the average threshold of human hearing at 1 kilohertz. But you can see that at any one region of characteristic frequency, there's a wide range of threshold sensitivity on the order of 40 dB. It turns out, as most of you probably know, that single auditory nerve fibers also have they have spontaneous rate. So even in a soundproof room, in the absence of any acoustic stimulation, auditory nerve fibers are discharging at different rates, and sort of surprisingly, many of them discharge at uniquely high rates. I don't believe there's any other place in the nervous system where fibers are discharging all the time, 24/7, even in the absence of the appropriate stimulus, at rates up to and even exceeding 100 spikes per second. So in this plot here, each one of these symbols is the spontaneous activity, spontaneous discharge rate in spikes per second of a different auditory nerve fiber plotted as a function of its characteristic frequency. It's obvious from this plot that regardless of which characteristic frequency region you look in, it looks like there's a bimodal distribution. There seems to be a cluster of fibers with what we call high spontaneous rates and another cluster of fibers with low spontaneous rates. And that's obvious also in the histogram here. There's a rate range around 18 to 20 spikes per second that is not very often seen, and that value sort of naturally divides this bimodal distribution. And it turns out that there's a very close correlation between spontaneous rate and threshold. So here on this plot over here, we have colorized the thresholds at the CF according to the spontaneous rate. So the ones in green here are those with high spontaneous rates, and the ones in the reddish tone are those with low spontaneous rate. And it's obvious that that's a very clear-cut distinction between high SR and low SR fibers. So look at it a little more systematically. Years ago, we decided to plot relative threshold versus spontaneous discharge rate. So to make these plots, because we had so many fibers in a single animal, we could quite accurately eyeball sort of the best thresholds as a function of characteristic frequency, and then for each fiber, express its threshold relative to the fibers with the best thresholds at the same characteristic frequency, i.e. fibers with the innervating inner hair cells in roughly the same region of the cochlea. So when you make that plot and you plot relative threshold versus spontaneous rate, you can see that indeed the relative threshold range of the fibers with high spontaneous rate is very compact, and it is very different from the relative thresholds of fibers with medium and low spontaneous rate. But it looked to us like there was something interesting going on even amongst this low SR group. And if we expand the horizontal scale here, you can see that indeed when spontaneous rate got below about a half a spike per second, it looked like there was a high probability that there could be very high thresholds. So based on plots like this, we proposed that the auditory nerve population should be divided into a high SR group, a medium SR group, and a low SR group, with the dividing lines being 18 spikes per second here and half a spike per second here. and with the relative frequency with which you would encounter them being 61%, 23%, and 16%. So it's interesting to ask whether the same thing is true in other species. Here are some data from guinea pigs. You can see that it also looks kind of like a bimodal distribution, and that bimodal distribution looks like it's maintained from apex to base, from low CF to high CF. If you do the same kind of plot of spontaneous rate versus relative threshold, it looked to us like the best place to divide the high spontaneous rate group off was 20 spikes per second, and the best place to divide off the low spontaneous rate group was at 1 spike per second. Well, how about the mouse? Oh, I'm sorry, the proportions are 73%, 15%, and 12%. So the mouse is not as clear-cut. There certainly is a relationship between spontaneous rate and threshold, but there's sort of more overlap, and it's not immediately obvious where to make the separation. But from a plot like this, it looks like it might be appropriate to divide them at six spikes per second. Despite the fact that this relationship between spontaneous rate and threshold doesn't look as much of a step function as it did in the cat data or even the guinea pig data. Much more recently, Birka Shrestha in Lisa Goodrich's lab did a single-cell RNA-seq analysis of spiral ganglion cells in the mouse, and then an unbiased cluster analysis, and concluded, based on the patterns of gene expression, that there were indeed 3 types of auditory nerve type 1s. We'll say more about that later. So what is it that gives rise to these differences in spontaneous rate? What do we know about the mechanisms underlying SR group differences? Well, years ago, we undertook a serial section electron microscopic study to look at this region between the habenula and the synapses. And this was done in cat. So in this image that I'm showing you here, it's a cross-section taken parallel to the basilar membrane of all these unmyelinated terminals of type 1 auditory nerve fibers as they traverse the neuropil underneath the inner hair cell. Here are the pillars here. Each one of these elements like this is an auditory nerve fiber. These darker things are mitochondria. And what we noticed in these reconstructions was that some fibers tended to have fairly large caliber and were full of mitochondria, whereas other fibers were much smaller in caliber and had many fewer mitochondria, and they tended to be spatially separate. That the ones on the medullary side were where one found the thinner mitochondrion poor fibers, and on the pillar side you were more likely to find the larger mitochondrion rich fibers. Because we had serial sections, we knew which fibers ended up having synaptic contact with the inner hair cells. Obviously, it made sense that if a fiber had high spontaneous activity, there would be higher metabolic demand. And so it made sense to hypothesize that these mitochondrion-rich fibers were those with high spontaneous rates, and these mitochondrion-poor fibers were those with low spontaneous rates. We had followed them all the way from the habenula to the synapse, and so we could see that there were some mitochondrion-poor fibers that never had more than a single mitochondrion in a cross-section, where others had several regions where there were lots of mitochondria. And based on this mitochondrial content, we divided them into mitochondrion-rich and mitochondrion-poor fibers. The mitochondrion-rich ones were larger in caliber, and the mitochondrion-poor were smaller in caliber. And indeed, they were morphologically polarized. So as shown in this schematic of the circumference of an inner hair cell, you can see here that the mitochondrion pore fibers are almost exclusively on the medulla, making synaptic contact on the medullar side of the inner hair cell, whereas the mitochondrion-rich fibers could be found anywhere around the inner hair cell circumference. I told you before we were able to intracellularly label single auditory nerve fibers with horseradish peroxidase, and so that allowed us to more definitively test the hypothesis, and it was shown to be the case. So here, each one of these points is a different auditory nerve fiber where we measured its tuning curve and its spontaneous rate, and then injected it full of horseradish peroxidase, traced it to its terminal, and in these light microscopic analyses of surface preparations, tried to estimate where around the inner hair cell circumference they were found. And so as you can see here, as predicted, the low SR and medium SR fibers were found exclusively on the medullar side of the hair cell. In this study, the high SR fibers were predominantly, but not exclusively, on the pillar side of the hair cell. So it really did seem like this morphological polarization was an organizational principle, at least in the cat. And I'm not going to show you the data, but we went on to show the same thing in the guinea pig ear. So we know, based on single fiber labeling, at least in the cat and the guinea pig, that this spatial polarization of the synapses around the inner hair cell is true in both of those species. So now I'm showing you again this cluster analysis of the gene expression patterns in spiral ganglion cells in the mouse. And this is another figure from that paper showing that, consistent with the idea that these three groupings have something to do with the three spontaneous rate groups, you can see that when Bricca Shrestha quantified the positions of these terminals of these different clusters along around the circumference of the inner hair cell, he found indeed that there was also a medullar pillar gradient. So it really seems like this general organizational principle and spatial polarization holds across mammalian species. Using the single fiber labeling approach, we went on to take this analysis to the electron microscopic level. We recorded from single fibers, measured their spontaneous activity, traced them to their terminals, and then cut up the material for electron microscopy. And it's possible to make this horseradish peroxidase label visible in the electron microscope. So the results of the analysis are shown here. What we're schematizing in these images is the active zone and the synaptic ribbon. So for example, this schematic here is as if we were in the inner hair cell looking down on the synaptic ribbon and the active zone, pre and post-synaptic active zones. And so this is the outline of the active zone in the red tone here, and the silhouette of the ribbon in the black and gray tones. So here is the total sample of fibers that we were able to trace and reconstruct: low SR, medium SR, and high SR. And to our surprise, we found the seemingly paradoxical result that low SR fibers tended to have larger active zones and more complex, longer synaptic ribbons with actually more synaptic vesicles around them than the high SR fibers. Much later, we showed that you can see signs of this gradient of synaptic ribbon size in confocal images. So here we show the CTBP2 labeling of the presynaptic ribbons along with Myo7A used to label the hair cells. These are from a guinea pig. This is actually a row of about 10 or 11 inner hair cells imaged from as a standard surface view, but then reprojected virtually so that we're looking along the spiraling axis of the organ of Corti, and it's very easy to see in this picture that the CTBP2 puncta, i.e. these presynaptic ribbons, tend to be larger on the medullary side than they do on the pillar side. This gradient is especially clear in the guinea pig. We haven't ever gone back to look at it in the cat. We've looked at it a lot in the mouse. And in the mouse, the gradient is there too, but as you can see, it's not as crystal clear. But the situation in the mouse is complicated because as schematized over here, the inner hair cells don't line up as well as they do in other species. They tend to be in this staggered position in many regions, essentially because the bottoms of the inner hair cell are too big compared to the region up here at the cuticular plate, and there's not room for them all to line up. At least that's my view of it. And so they stagger in position. And so in this image here, this one of the image inner hair cells might be reflected out this way, and another one might be reflected in more toward the pillar. And that staggering of the inner hair cell position complicates the analysis of the medullar pillar gradient in the mouse. So now I'm going to turn in the next few slides to work from Tobias Moser and his group. He has spent a lot of time with in vitro approach to try to understand what it is that causes low spontaneous high threshold fibers to have low spontaneous rates and high thresholds. And here's one nice study from that group from 2016 where they used immunostaining to look at the voltage-gated calcium channels in the presynaptic membrane and calcium imaging to look at calcium influx when they depolarized the inner hair cell with these patch clamp electrodes. And what they found, again, perhaps seemingly paradoxically, is that these putative low SR synapses on the medullar side showed more calcium influx than on the pillar side and more voltage-gated calcium channels than on the pillar side. In a subsequent study, they showed that there was a difference in the membrane voltage required to activate a calcium influx on the pillar side versus the medullar side. So these are fluorescent traces of calcium influx. Here's one pillar synapse and here's one medullar synapse, and you can see the different membrane potentials here, and you can see that there's signs of calcium influx at minus 49 millivolts on a pillar side synapse and not till almost 10 dB, 10 millivolts higher on the medullar side synapses. So they proposed that there are different flavors of voltage-gated calcium channels, and that the ones on the pillar side are activated at lower membrane voltages than the ones on the medullar side. Putting all that together, schematizing these different flavors of voltage-gated calcium channels with these different colors, they proposed that the active zone on the medullar side contains these channels that require more membrane depolarization, but there are more of them and they're more diffusely organized than on the pillar side where they're more compact. And that is reminiscent of what I showed you before, where we had seen this paradoxical result where it looked like the low spontaneous rate active zones were bigger than the high spontaneous rate active zone. So this and other studies from the Moser group and others have shown that there are clear differences in the presynaptic behavior of these synapses at the pillar side versus the medullar side. We've recently shown that there are also interesting postsynaptic differences. So this confocal image here is from a guinea pig. We're looking down on the osseous spiral lamina. So these are the myelinated ends of the type 1 fibers as they approach the habenula. And this is the synaptic region, the inner spiral bundle, and the tunnel spiral bundle here. The labels that we've used here are cell mask to, it's a lipophilic fluorophore that labels myelin very nicely. And synaptic ribbons are shown here in white with CTBP2 staining. But what's most relevant here is this contactin-associated protein, Casper, which is a marker of nodes and heminodes in green. And the important point here is that there are some heminodes where we believe the spike generator is, where the myelin ends that are quite far from the habenula, and there are other heminodes that are a lot closer to the habenula. And in this image, you can see that the ones that are farther from the habenula tend to be thin, and the ones that are closer to the habenula tend to be thick. So what we believe is going on here is schematized here, which is that the heminodes of the high spontaneous rate fibers tend to be closer to the synaptic region than the heminodes of many of the low SR fibers. So there's a spatial gradient in the osseous spiral lamina. It also turns out there's a bit of a spatial gradient here such that the low SR synapses, the medullar side synapses, are farther away from the habenula than the pillar side synapses. We've known from prior studies that the caliber of the high SR fibers is thicker than the low SR fibers, and so the argument is that there are more space constants between the synapse and the spike generator for low SR fibers than there are for high SR fibers, so excitatory postsynaptic potentials will be attenuated more on the way to the spike generator for low SR fibers than for high SR fibers. And we've argued that that difference, that postsynaptic effect, could very well be an important contributor, as well as the presynaptic differences, to the low spontaneous rate and high threshold of the medullar side versus the pillar side fibers. These are some of the data where we've tried to plot the positions of the heminodes from the scala vestibuli side to the scala tympani side, and the positions of the synapses from the medullar side to the pillar side. And you can see that there could be a very long distance from a low SR heminode to its synapse, and there could be a much shorter distance from a high SR heminode to its synapse. We have evidence from prior studies in cat and guinea pig that the unmyelinated terminals, as I said, of high SR fibers are thicker than low SR fibers. And I show you in the next slide the peripheral axons here of a slice through the osseous spiral lamina, and you can very clearly see that the terminal ends of the myelinated peripheral axons of the putative low SR fibers on the scala vestibuli side are smaller than those on the scala tympani side. Okay, so the last chapter, I'm going to say just a little bit about the basics of stimulus coding, with the important take-home message that one of the unique features about auditory nerve fibers is that they carry information both in terms of their average rate of discharge as well as their synchronization with the individual cycles of stimuli, their periodicity. So here in this classic illustration from a Kiang chapter from many years ago, he shows the idea that auditory nerve fibers are tonic responders. So here is a very long time course of 15 minutes. For 5 minutes, there's no stimulus. For another 5 minutes, there's a tone that's on that excites the fiber. And then there's another 5 minutes when there's no stimulus and you're just looking at spontaneous activity. And these are spike traces as they would appear on an oscilloscope screen. It's obvious looking at these spike counts where each point is the average discharge rate over a 100 millisecond time period. It's obvious that when the tone is on, the spike rate is higher than when the tone is off, and that high spike rate, as far as we know, is maintained essentially forever. Here we only show it for 5 minutes, but they're apart from a slow adaptation, which we'll talk about in a minute. The fiber responds for as long as the stimulus is on. But you also note from this interesting plot the high variability in the spike rate when you're looking at little samples of about 100 milliseconds. The auditory nerve response is highly probabilistic. And so what you can also see from a plot like this is that there are a number of individual 100 millisecond time samples where the discharge rate in this one fiber is actually higher when the tone is off than when the tone is on. So that to me is one of the obvious reasons why it's good to have lots of auditory nerve fibers per inner hair cell, because that allows the central nervous system to average responses over a number of fibers so that it can, in a very short period of time, get an idea of the average rate difference that's so obvious when you look at the data this way. So I pointed out this slow adaptation here. This one looks like it has a time constant on the order of tens of seconds. These very long time constants have not been very well studied, and their underlying basis is not very well understood. What's been a lot better studied is the post-onset adaptation on a much shorter time course, as seen here when we look at the discharge rate as a function of time after the onset of a 50 millisecond stimulus. So auditory nerve fibers show a very prominent onset discharge rate, a exponential type decay to a steady state that isn't reached for a few tens of milliseconds. You can also see that after the tone burst is turned off, it looks like the spontaneous rate is depressed for a while before it recovers back to its steady state rate. It's clear that this kind of post-onset adaptation is not present, at least not to the same degree, in inner hair cell receptor potentials, as shown in this classic plot of receptor potential as a function of time for 1 kilohertz, 2 kilohertz, 3, 4, 5 kilohertz stimuli. So our understanding, and there's plenty of evidence for this that I won't go into, that this post-onset adaptation is a synaptic event. It is constrained by the synapse. It's constrained by what I pointed out before, which is that at this synapse, the only way this auditory nerve fiber is communicating with the inner hair cell is by this synapse, at which there are only a couple hundred synaptic vesicles. It's actually pretty amazing that with this small number of synaptic vesicles, you can maintain a spontaneous discharge rate of 100 spikes per second, 24/7, to say nothing of these much higher instantaneous rates. It's obviously an incredibly dynamic system. Adaptation also contributes to masking, but low SR fibers are more resistant in a very interesting way. And I'm going to illustrate that in the next few slides. So when you're recording from a single auditory nerve fiber, its tuning curve is shown here, a high spontaneous rate fiber and a low spontaneous rate fiber. If you put a tone at the characteristic frequency and raise the sound pressure level, you can see that discharge rate only rises for a very small number of dB. There's a relatively narrow dynamic range over which discharge rate moves from spontaneous to saturated rate. Low SR fibers in many species have been shown to have a slightly larger dynamic range, but for all fibers, there tends to be saturation at high discharge rate, and this saturation phenomenon is also constrained by the synapse. And it's obvious that having threshold range increases the dynamic range to tone bursts in quiet, but what I think is most interesting about low spontaneous rate fibers is what happens when you add noise. So over in this next set of view graphs, we look at this stylized rate versus level function of a high spontaneous rate fiber, like we showed over here. And this would be its response to tone bursts in quiet, as we showed over here. But in the darker color, we show what happens to this rate level function when you add a continuous noise. If it responds to the noise, then it obviously increases the rate even when the tone burst is at low sound pressure levels. But what's interesting is that at high sound pressure levels, it actually decreases the rate of the fiber's response to the tone burst, and that is because of adaptation. The presence of this continuous noise fatigues the synapse. It gets rid of all the readily releasable vesicles, and so the rate level function to the tones is compressed. There's a higher rate here and there's a lower rate here. And years ago, Eric Young and colleagues looked at this compression phenomenon, which they expressed as the ratio between the spike difference between here and here in the absence of noise and the spike difference between here and here to the tone burst in the presence of noise. And this ratio of A to B, this compression of the rate level function, is very different in high spontaneous rate fibers, which are very easily compressed, and low spontaneous rate fibers, which are much less easily compressed. And our understanding of this is it's simply because their thresholds are higher, they are driven to a lesser degree by moderate levels of noise, and therefore they are compressed less. So my view is that one of the true importance of low spontaneous rate fibers is really revealed in a noisy environment, perhaps much more so than in a quiet environment. So the very last couple of points that I want to make get to the idea that in addition to coding information as an increase in average discharge rate, auditory nerve fibers can code information in their periodicity. So here I show a spike train as it would appear on an oscilloscope screen. And what we're going to show are histograms of the interspike intervals. So instead of looking at the times of occurrence of spikes with respect to an external stimulus, we're just looking at the intervals between successive spikes. And we're going to show interval histograms in the spontaneous region, in the tone-on region, and in the spontaneous region again, and here's what they look like. So in the absence of a stimulus, these interval histograms, when they're plotted on a log scale here, show this exponential decay of intervals. However, when there's and that has significance, it tells us that the intervals between spikes are completely random, and that's very important. But in the present context, what I want to point out is that when the tone is on, there's obvious periodicity in these interval histograms. And if you count the number of peaks here, there are 12 peaks, and those 12 peaks occur over 50 milliseconds. There's a 4.2 millisecond period, and that's reflecting the fact that this was a 240 hertz stimulus. So this fiber is not responding every 4.2 milliseconds, but its spikes tend to be separated by integer multiples of 4.2 milliseconds when there's a 240 hertz stimulus. This is another way of looking at that. Here's a sinusoidal stimulus. People have constructed things called post-zero crossing histograms, where we reset a clock to zero every time the stimulus crosses zero, and you can see the periodicity of the spikes as the sound pressure gets above threshold, and you can see that reflected in this periodicity in the positive zero crossing histogram. From these histograms, people have extracted numbers that describe how synchronized these fibers are, and without going into the details of what that number is, it's called a synchronization index, and it goes from 0 to 1, and the higher the number is, the more synchronized the spikes are with the stimulus. This is a summary plot from a paper by Don Johnson a number of years ago, where as a function of tone frequency, he shows the synchronization index for stimuli well above the maximum synchronization index as a function of stimulus frequency. And what you can see is that this periodicity is only visible for low frequency stimuli, that is below 4 kilohertz. But one of the key points about this is that this axis here is tone frequency, not characteristic frequency. What I mean by that is that for any fiber, whether it has a low characteristic frequency or a high characteristic frequency, if you get the stimulus within its tuning curve, there will be a high degree of synchrony. This fiber that has a characteristic frequency of 16 kilohertz at 70 or 80 dB is responding very synchronously to low frequency tones. And the take-home message from that is that even for stimuli in the speech range, i.e. at low frequencies, there's a tremendous amount of information carried by high frequency fibers, even extended high frequency fibers. And we think that that makes them particularly important, and is part of the reason why, as we lose our high frequency hearing, we have more trouble understanding speech, especially in noisy environments. So that's the end of my story. I want to, I've talked about too many studies over too many years to make specific acknowledgments. So I'm just putting up pictures of the whole Eaton Peabody Lab crew in 1984 and 2024. And I've had incredible help from many of them over the years. And here's my funding sources. So I'm just going to put up my conclusions and leave them up there for you all to read, and stop there and see if there are any questions that I can answer.
Thank you so much, Charlie. That was wonderful. We do have a couple of questions in the chat. There was one that was asked early on that was immediately answered. It was relating to why you used cats when they have more synapses compared to humans, and you immediately answered it. So that's awesome. We've got one from Brian Moore. If you'd like to unmute, please feel free to do so, Brian, and you can ask your question, or I can state it for you.
Yeah.
There you are.
So, Charlie, I think I asked you this a long time ago, and I'm asking it again now. What's known about the distribution of spontaneous rates and thresholds in humans?
Right, nothing.
Same answer.
It's easy to answer.
Yeah, I mean, are there any, is anything known from anatomical work, for example? Are there any analogues in the anatomy and staining that look like the animal data?
Well, we've tried to me, the best way to get at that would be this one, looking at the peripheral axons. The problem is that in postmortem material, there's a lot of swelling and bubbling and distortion of all this stuff, and so it's tough to do. I mean, if we could ever get a young human specimen with very, very short postmortem time, I mean, I think you could at least look at this scala vestibuli, scala tympani gradient of axon caliber and get some ideas about it. Based on the work on these gene expression differences, there are now markers that allow us to differentiate the different spontaneous rate groups. And the problem is that the robust marker for the low spontaneous rate fibers is not available as an antibody. It has to be studied with messenger RNA expression, and there are a number of people working on trying to get that technique working in human postmortem material, and it's obviously challenging because of the postmortem effects. So there are ways to get at it, and people are thinking about it and continuing to work on it.
Thank you.
I have a follow-up question, Charlie. Is there any way of potentially being able to look at the mitochondrial content in EM tissue, or is the swelling just too difficult?
Yes, I think that might be doable. I mean, I think if you had a good, short postmortem, reasonably well fixed human specimen, and you did the serial section reconstruction, that you could get some insight that way.
Okay.
So yes, that's another way. It's even more challenging because it's a longer distance from the habenula to the synapse.
Yeah.
So it's like 2,000 sections instead of 1,000 sections.
Maybe with
If some, yeah.
Yeah.
Right.
I had a quick question about that as well, with respect to, because there are fewer numbers of synapses in human, do you think that, so first of all, are there more inner hair cells in human
Yes.
compared to cat? And so do you think that there might be similar numbers of fibers and they're just distributed differently on each?
Yes, that's true. I mean, there's a remarkably constant packing density of inner hair cells from humans to mice, which is about 10 micron on center spacing.
Okay.
And so a human cochlea is like 35 millimeters long, and a cat cochlea is like 25 millimeters long. And yeah, if you multiply out the total number of fibers in humans, in a young human's probably about 40,000.
Okay.
And in a mouse, it's probably about 40,000. I mean, in a cat, it's probably about 40,000. In a mouse, it's a lot less. So
Yeah.
it doesn't stay constant as you go to smaller and smaller mammals.
Okay. All right, I'll get back to the ones in chat. Mehmet, I believe you had a question. Are you still here, and would you like to ask it?
Yeah, of course. Thank you. Hi, Dr. Liberman. Great presentation. Thank you. So right at this slide, actually, you showed us type 1A, B, and C is separated very well and nicely in three different groups. But when I do image staining for type 1A, B, and C, I see some overlap. Sometimes single SGNs express more than one. In some occasions, they express all three of them at the same time. So I just want about that. Like, have you come across to that before, or how do you interpret it?
Well, yeah, you're not alone in that observation. I have not myself done any of those labeling studies, but I've seen the work and seen the published and unpublished work, and you're absolutely right. There's overlap, and it's a gradient. It's not all or nothing. The high spontaneous rate fibers are richer in calretinin, but it is not the case that there's none in the type 1B and 1Cs. They're all gradients, and you can see that there's some overlap here, and these kinds of plots here show the best guess of compressing into 2 dimensions a multi-dimensional space with gene expression of lots of different genes. So yeah, they are gradients. They're not
Thank you.
all or nothing.
Okay, I know we're a little bit over, but we've just got one more in chat that I can see. So, Anesia Banerjee, are you still here? Would you like to ask your question?
Yes, sure. Thank you so much, Charlie, for this fantastic talk. I guess I was wondering how the differences you mentioned in the length and the diameters of the fibers would affect the information propagation when the fibers are stimulated in case of cochlear implants. Because I understand that there is there is much limited spontaneous activity because of how the synapses are not in the game really. It's direct stimulation with the electrodes, but there is still this physical differences, and can that limit any kind of information propagation and result in some behavioral deficiency? Do you know anything about that?
Right. Well, when the hair cells are gone, there's absolutely no spontaneous activity. That is pretty clear. And in most cochlear implant users, based on human temporal bone analysis, although there may be lots of spiral ganglion cells surviving, many of them have no peripheral axons. And I think my own view is, and I think other people agree with it, and there's other evidence for it, that doesn't keep them from being electrically stimulated. In fact, years ago, when we were recording from the auditory nerve in severely noise-damaged cats, we created regions where the organ of Corti was completely destroyed, and in those regions, the peripheral axons were degenerated. And when we recorded from the auditory nerve using shocks at the round window as a search stimulus, we found many fibers that had no response to sound and no spontaneous activity, but were electrically excitable. And when we looked at their shock latency, it didn't look any different from the fibers that were still acoustically responsive. So that always led me to believe that it's just as easy to stimulate the cell body directly as it is to stimulate a fiber that has an unmyelinated terminal and a myelinated peripheral axon, and that it doesn't compromise their potential performance in a cochlear implant.
Thank you.
We do have a couple more questions in the chat, but in the interest of time, I think we're going to have to call it there. So thank you to Charlie for your wonderful talk, and thank you all for your engagement. I think if you'd like to direct questions directly to Dr. Liberman via email, that should be fine. And just a reminder that this will be posted online for future viewing at the ARO YouTube channel. So thank you very much, everyone, for your attendance today. Have a great day.